in the JSS College of Pharmacy, UT, and subsequently promoted as associate professor. And presently, sir is the head of the department of pharmaceutical chemistry and vice principal for that college since December 2013. And sir is a life member of APTI and member of editorial team for various international journals. And sir is working as reviewer for various international journals and many conferences and seminars, as sir has worked as a PC member, and sir was also in yeah, sorry for the disturbance. Am I audible now? Yeah. So sir has participated in many conferences and seminars as PC member and invited speaker. And sir's main research interests are development of antibacterial agents, molecular modeling, drug design, custom synthesis. And sir has published more than 41 research papers in peer-reviewed journals. And sir is involved with sponsored projects of national funding agencies and the administrative work of college. And in this capacity, he coordinated with academic council and various academic departments of university and supervising academic programs. And with this short note, I'd like to introduce today's speaker, Dr. Absal Azanser. Welcome, sir.
Yeah, thank you. Thank you very much for the nice introduction.
Yes, sir. Over to you,
allow me
sir.
to share the screen.
Yes, sir. Yes, sir. You can share, sir.
Okay.
Once you share, sir, we'll see here.
You just allow it. Yeah.
Yes,
so
sir.
What very good afternoon, all the participants. It's my pleasure to talk something about this molecular modeling tools. Okay, useful tools. You can say very useful tools. And in this new world, or new whatever research you are doing, there it is very, very helpful. And what I'm saying, most of you are from the academic background, some of you are not from the chemistry background. So I'll go very low at the first in the beginning. I'll tell you something about the chemistry part of the protein. Okay, without knowing the protein, it is not possible to do the molecular modeling or any drug design, okay, approach. You cannot use. So here, just I'll share one screen and okay, because many of you are not from the chemistry background. So let us see here. Yes. My slide is visible?
Yes, sir, it's visible, sir.
Yeah?
Continue, sir. Please continue, sir.
No, you look at very short. I'll tell you in short. I'll tell you the proteins are, as all of you know, that is made of the amino acids. Okay, amino acids are linked together by the amide linkages. Okay, amide linkages. Now you can see here. 1 minute. Yeah, amide linkages. Now this amide linkage here you can see at this linkage there is a C alpha carbon. Can you see here? CO. This is the CO. This is the NH2, and this is the C alpha. Over the C alpha, whatever the group is there, this group is known as side chain. Known as side chain. So when you see whatever the proteins are there, the protein may have very long chains are there. You can see these are 25 amino acids. So there's a two terminal. You have the amino terminal, you have the carboxyl group terminal. Two terminals are there. So any protein you take, because you are in the molecular modeling, you're going to use the protein. Whether you're taking from the protein data bank and anywhere. So those all proteins have two ends. Okay, amino end and carboxylic end. Okay, two ends are there. Now you have to be very careful. Okay, when you are handling the protein. Okay, if you know well, okay, if you know well, then it is then your result will be good. Okay, otherwise you will not get a good result. Okay, now you can see here this 1 minute. This is disturbing. Yeah. So now here you can see this amide linkage. Okay, amide linkage C alpha, and this is C alpha, and this is your side chain. Now this is another photograph, which is showing very simple photograph I taken. Amino acids are there, C terminal and is there, and any amino acid have amino group, carboxylic group. So amino acids, okay, amino acids are generally unilateral code or trilateral code, you can call it. Okay, so alanine, arginine, generally in the molecular modeling, we use the trilateral code. So alanine, just arginine, ARG. So everywhere in all literature, you see three-letter code will be there. Okay, generally it is written. Some of the literature, they use unilateral code. Okay, unilateral code. So this is one important aspect of the amino acid. Second thing, now when you take any protein, you can see here this is a long chain. Suppose it's a long chain means now this here C alpha here you can see this is the C alpha. C alpha, okay. Here you can see this is the what type of group here? It is your lipophilic group. It is your because CH3 is okay, isobutyl group. Isobutyl group is there. Okay, here you can see the phenyl ring is there. Okay, here you see this is the here now your amide linkage is there. Amide group is there. So now the protein nature depends on depends on the side chain. Whether the amino acids, whatever the amino acids are, they are polar or non-polar. Okay, it depends on that. It depends on that. Okay, so now this amino acid, we have to know, we should know what type of amino acids are involved in the binding of the drug or binding of the ligand. Okay, binding of that. That's why you have to study. Okay, whenever using any protein from the protein data bank, download the literature and see what is the catalytic pocket residues are there, and those residues, what is the nature? What is the nature? It may be hydrophobic, it may be the lipophilic, okay, or it may be the it may be the ionic, or it may be the polar group. So the whole your design of the molecule depends on the nature of the amino acids in the catalytic pocket. This is one aspect. Second thing, then when the protein is there, so this is your backbone. You can see this is long. Backbone may be very, very long. So and this is a side chain. So there's a fluctuation in the, there's always fluctuation in the your physiological condition. Proteins are always in the dynamic state. Okay, surrounded by water. Surrounded by water. It is always in the dynamic state. So there is a flexibility. There's a flexibility. Okay, in your backbone. This is the backbone. There's a flexibility in the backbone. There's a flexibility in the side chain. It's in that side chain. This flexibility matters a lot when you're designing a molecule. When you're designing a molecule, okay. These amino acids. Now you can see we have to be very, very careful when selecting, when you're looking into your catalytic pocket. Now you can see alanine, asparagine, cysteine. Okay, these are all you can say these are the neutral amino acids. These are the neutral amino acids. Okay, and then here you have the acidic amines, aspartic acid. Then okay, this is your acidic amino acid. Then you have here you see arginine. It is also having carboxylic group. So acidic in nature, and the basic in nature histidine is there. Okay, histidine is there. Then you have glutamic acid is also there. So these amino acids may be neutral, amino acids may be acidic, amino acid may be basic. So you have to look into the catalytic pocket, what type of amino acids are present in the catalytic pocket. No, not only it ends here. Now you see that then if you look into suppose in the catalytic pocket you have the tyrosine. So this phenyl ring here, okay, L-tryptophan, phenyl ring here, okay, or you see L-phenylalanine, phenyl ring here. This plays a very important role, okay, in hydrophobic interaction. Hydrophobic interaction. So if these benzene rings are there near in the catalytic pocket, we can use these benzene rings also, these hydrophobic residues also to develop the inhibitor, to develop the potent inhibitor. Or if suppose the carboxylic group is there or basic histidine is there, then also we can use it. Now this is a very peculiar amino acid. You can see L-histidine. Now here it will form the hydrogen bonding. It will accept the hydrogen bonding. Okay, except the hydrogen bonding. This is also both things are happening. So histidine plays a very, very important role. L-tryptophan, okay, tyrosine, L-phenylalanine. These all plays a very, very important role. Okay, in design of any molecule. Okay, so these things, basic things you have to keep it in mind when you go ahead with the design of any molecule. Now I'll come to next. Now these amino acids, you can say there may be primary structure. Primary structure, you know, this is the chain, simple, it tells about the sequence. Then it may be secondary structure, maybe alpha helix, it may be plated structure, beta sheet. Okay, then it will fold, after folding, it will form the tertiary protein structure, and then again it will form the quaternary structure. Quaternary structure. Now, these structures are very, very important. So any amino acids, any protein, you see these are stabilized by so many interactions within. Even the protein itself is stabilized, okay, by these linkages. Now you can see it may be stable due to the hydrogen bonding between the different residues. There may be hydrophobic interaction, okay, isopropyl group, hydrophobic interactions. Okay, there may be disulfide bridge and ionic bond. Now, suppose you observe any this ionic bond or disulfide bridge and hydrogen bonding, don't try to disturb that. Okay, when you're selecting any protein, don't try to disturb that, because if you remove this hydrogen bonding, it may become unstable. It may become unstable, and then problem is that the conformation of the protein will change, and you will not get the good result. You will not get the good result. This is one thing you have to keep it in mind. Okay, so these interactions are important for the stabilization of the protein. Okay, so this is, we have to keep it in mind. So now you can see, this is another diagram. You can see primary, secondary, tertiary, quaternary structure. So this is the basic thing I told you about the protein. So if you have any question, you can ask. Otherwise, I'll proceed. Okay. Now here next. Now I'm just going to the now. What are the ways to identify the lead? What are the ways? How do you go ahead? Suppose you have any protein. You have any protein. What are the different ways? What is the best way? Now that nowadays companies they use fragment-based drug design. So fragment, they crystallize the protein and they go for the NMR spectroscopy, and then they develop the molecule. They develop the molecule. But we cannot afford. Okay, academic level, we cannot afford. So how we can proceed? How we can proceed? So one way is, okay, so simple is the molecular docking. You can go ahead with the molecular docking. Okay, molecular docking. Molecular docking is not only the solution. It is not only the solution. Okay, it is not only the solution. Now I'll share the screen. So can you see this one? Hello?
Yes, sir.
Can you see this screen?
Yes, sir. Yes, sir. We can see you. Yes.
Yeah. So now just I'll import one file and show you. Okay. So now how you can, I'll start with the docking. Okay. Docking protocols, there are so many you will be knowing also. A little bit I'll tell here. Okay. A little bit I'll tell here also. 1 minute. Let me. Yeah. This you can do by using the free software also. Okay. You can use and you can do this work. So now you can see, okay, here we have, I have uploaded one protein. Now let us see how to analyze this protein. Okay, how to analyze this protein. So just I'll change the color, okay, and then it will be visible to you. So okay, and 1 minute, I'll take the ligand also there, and then yeah, now it is. Is that visible now?
Yes, sir. Yes, sir.
Yeah, yeah. Now, yeah, that's here. So just 1 minute. Let me optimize it. Now you can see this is the co-crystal ligand. This is it gives a lot of information. So what you have to do when you are going for any docking study, okay, you have to first start with the co-crystal ligand. How it is binding. You can see this is the co-crystal ligand. Okay, so how it is binding here, okay, and how we can improve the affinity of this inhibitor. Okay, now literature will tell you what are the binding residues. Now I just generate a catalytic pocket here, shape of the catalytic pocket, okay, then it will be easy for you to understand. Okay. So now generate binding site surface. Okay. Okay. This is you're using the academic version. This also you can download and you can use. Now you can see this is the catalytic pocket. Okay, of this. Okay, this protein. Now you can see how the ligand. You can see ligand is or inhibitor is within the catalytic pocket. It is well fitted into the catalytic pocket, isn't it? You can see it is well fitted in the catalytic pocket. Okay, and this catalytic pocket, now you come to know what type of molecule you are going to design. Okay, so this is a flat molecule. Very simply, you can say it should be a flat molecule. If you some bending you do, then it will not fit in the catalytic pocket and you will not get the activity. Okay, you will not get the activity. Okay, so this is the prime thing you have to. Okay, you have to see into that. So the catalytic pocket, you can see it is hollow, okay, long one, a straight one, okay, and then your inhibitor, whatever is there, okay, it should not be twisted one, okay, then only it will fit. Second thing, after looking into that, now you can see the red color region. This is a hydrogen bond acceptor group. Can you see this is a CO group? Is it visible? Carboxylic group.
Sir, yes, sir.
This is the carboxylic group. Okay. Here you have amino group. So blue color donates hydrogen bond donating region. Hydrogen bond donating region, and red color shows your hydrogen bond acceptor region. Okay. Now you analyze the pocket. Okay, you analyze the pocket and you can see what changes you are going to make, okay, to improve the activity. Now I'm seeing this ring, you can see this is coming out of this ring is coming out of the catalytic pocket. Okay, it is coming out of the catalytic, it is not fitting well. So I don't think it is required here. In place, if you put some other group here, and then suppose it forms, okay, any hydrogen bonding with this, okay, carbonyl function here, then definitely it will improve the activity. It will improve the activity. So here on this benzene ring, one we can go for the substitution, we can different substitution, and then we can make the inhibitor more active. Okay, understand my point? We can make inhibitor more active. So this is one thing here. Now you look into the other region, what is happening there. Now here you can see there is a chlorine. Okay, sometimes chlorine very important. It will form, okay, halogen bonding. Sometimes halogen bonding. Now here you see sulfoxide group is there. Okay. Then you have in this region you can see. So here amino acids, different amino acids are there. Here also carboxylic group is there. So this if we can use these groups, okay, we can go for the substitution, okay, on the ring system, or we can replace the ring system here to improve the activity. Okay, so that more hydrogen bonding and the molecule shape should not change. It should be a straight molecule, and then okay, by substituting different groups, we can improve the activity. This is one approach which you can use. This is one approach which you can use for design of molecule. Okay, and not only this one, even you can start. This is a simple thing I'm telling. So when you're going for the molecular docking, you dock suppose 100 of compounds if you dock, and analyze one by one and see which compound is fitting well in the pocket. Okay, select that compound. Okay, select that compound, and then substitution you go on changing all over the ring system. Okay, and then again you dock, redock, then you will come to, okay, the binding affinity. Okay, it will improve. Binding affinity will improve. Now you can suppose after doing all these exercises, you came to about 25 molecules with high binding affinity. So after doing all these exercises, okay, so these 25 molecules, then whether these 25 molecules are good enough, how you judge? Then you have to go for the binding free energy calculation. Okay, there are two ways. Some software directly you can calculate by MMGBS approach, and some software, like if you're using the free software, okay, then and then Gromec and all, then by after doing the your dynamic simulation study, you can break up the energy you can find. Okay, so what is the contributing binding free energy? But that will take more time. That will take more time. But binding free energy gives the accurate one. Okay, accurate result. Accurate result. Okay, this is having more binding affinity. So okay, so now suppose that you are getting the binding free minus 70 kilocal per mole. That means compound is very stable in the catalytic pocket. Sometimes this docking will give the false result also. Okay, then how to validate it? Whether your docking is correct or not. Okay, then what you have to do, whatever the co-crystal ligand is there, that you take out from the catalytic pocket. Okay, you remove it, and again, okay, after preparing the protein, then you have to redock it. And then you superimpose the co-crystal structure with your docked structure. If both the structures are superimposing, you understand my point? Hello, anyone of you?
Yes,
Okay.
sir.
Yeah. So now suppose that you have co-crystal structure. So remove the co-crystal, take out the break up the co-crystal structure, remove, and then the co-crystal structure. Okay, and then again, okay, prepare the ligand and the protein you prepare, and then dock it again dock it, and then you just again take the co-crystal structure and then go for the superimposition of the dog pose with the co-crystal structure. If both the structures are superimposing with less RMSD, means less than 1, it is better if it is lower, 0.7, 0.6, okay. If it is better, then you can validate that your docking is good. Docking is good. Whatever the protocol you have followed, it is good. Okay, so you have to validate the docking. Sometimes what happens, suppose 25 ligands are there. So what you have to do, you use two softwares. Okay, in one, both software you cross check. Okay, whether it is showing the same result. Okay, it is showing the same result. Okay, so that also is one of the validation process. So validation is must for docking. Validation is must for the docking. So this is one approach. And when you're going for the docking, okay, another thing is that protein you have to prepare very carefully. Okay, energy minimization should be there. Okay, you have to minimize the energy. Okay, without minimization of energy, that it will not be stable. It will not be stable. So you have to minimize the energy of the protein, and then only you use it. So minimization of energy is one important aspect when you are preparing the protein. Now, why I'm telling this one? Because now I'll show you one video. Okay, I'll show you one video. Now you understood this one, why it is important here. Okay, so this docking, what two type of validation? One is the cross validation, means you use two type of software and check. Another is the your co-crystal ligand and your dock pose. You just superimpose and check. So this is another approach. Okay, this is must. If you do it, then you are satisfied. Okay, my docking protocol is good. Okay, so this is done again. So it will give some of the docking. Okay, software will give so many poses. So which pose to take? And the question arises. So then you have to look into the energy. So which pose is having minimum energy? So based on the energy you can take, you can select it. Now another problem with this, your ligand conformation will change, protein conformation will change. Okay, this is in constant movement. This is in constant movement. Okay, constant movement in the, okay, then you can say it is a dynamic process within the body. Okay, so protein is always moving, always moving. Now here, when the protein is moving, so what will happen? I'll show you. So now one more thing you have, yeah, 1 minute. One minute. let me generate a ribbon. Now you can see. Now you can see this protein is having alpha helix. Okay, one more thing I'll tell you here. Protein is having alpha helix. Protein is having, this is the beta sheet. This is the beta sheet. It is the alpha helix. Okay, alpha helix. And then you can see there's the loop. There's a loop here. So the loop region, sometimes what happened, the inhibitor will go and bind in the loop region. Okay, so that you have to look into the flexibility there. What loops are highly flexible. Loops are, you can see here very big loops are there. Okay, so apart from the alpha helix and beta sheet, there's a large number of loop in this protein. So definitely this protein is highly flexible. So now this we have to see, if you run the molecular dynamics, then you will come to know how flexible it is, how it is changing the conformation, okay, and how we can improve the activity. That I'll show you afterward. Okay, now here, so any protein will have alpha helix, it will have beta sheet, it will have, okay, loop. So loops are also very important. Okay, loops are also very important. Sometimes the inhibitor will go and bind there in the loop region. Okay, so we cannot omit that one. Then the conformational change in the alpha helix. Okay, you have to observe the conformational change in the beta sheet also during the simulation studies. So these things are also very important. Now I'll show you and I'll come back to this again. Okay, I'll show you one more slide here. Okay, 1 minute. Where it is, 1 minute. Just let me open it, then you will understand what is the importance. This we generated after the dynamic simulation, okay, and then I'm just showing you so that you can understand. What is the importance, okay, of this, okay, protein. How we can control the protein. We can control the. I'll share it. 1 minute. Can you see this one?
Yes, sir. Yes, sir.
Is it visible?
Yes, sir. It's perfectly all right, sir. We can see
Yeah.
everything.
Now you can see this protein is highly flexible, isn't it? And the ligand is also flexible. Now you can see this ligand is between this loop region. Okay, it is there in the loop region. Okay, so this inhibitor is in the loop region. So that's why I'm telling this. This is very important. Okay, this is very important. You have to look into the protein. Okay, where it is binding, what are the amino acids there. Okay, what is the catalytic pocket shape? So based on the shape, based on the residues, nature of the amino acids, we can design the inhibitor. Okay, this is one photograph I have. Second one I'll show you, that is also very important. Okay, this you can see one more is there. Yeah. This is, I'm telling, practical aspect so that you can use it. Okay, you can use it. Now this we have generated. Okay. Is that visible now?
Yes, sir. It's a visible, sir.
Hello?
Yes, sir. Hello, sir.
Visible?
Yes, sir. Yes,
Yeah?
sir. Yes, sir.
Visible? Now you can see how flexible this is. Okay, this is with the surface. The catalytic pocket, you can see it is very narrow, and inhibitor always moving inside. Okay, and this is also, you can see this is surrounded by the loop, isn't it? You can see here is the loop. Okay, you can see here is the loop, here is the loop, here is the loop. Okay, so this is surrounded by the loop. Okay, highly flexible protein. An inhibitor is also changing the conformation. So this is the one of the thing we have to keep it in mind. Now, how to address this issue? Okay, one protein we cannot control, conformation will be there, change will be there, resolving the constant movement. So suppose your inhibitor is not showing high activity, then what you can do? Okay, then now you can see what I'm seeing here. There is a rotation here. Okay, so there's a lot of rotation, single bond rotation, single bond rotation. That you can prevent it. Single bond rotation, you can prevent it. Suppose you take it in the cyclic form. If you put it in the cyclic form, you can see it is. You see, conformation is not stable. It is not stable. It is constantly moving. Okay, constantly changing the conformation. Okay. Now, if it's changing the conformation more, the most of the time, okay, it will not be stable. It will not be stable. So another way is that. So how to do it? So now this molecule make a rigid molecule. Try to make rigid molecule. Okay, when it is not stable. If you rigidify it, then what will happen? Then what will happen? It will form a stable interaction. It will form a stable interaction within the catalytic pocket. So that is another way we have to look into. Another way we have to look into. So this is about your how to use the catalytic pocket. Then another thing comes. Suppose you have some set of molecules, okay, from the literature, or you want to develop your own molecule, okay, with high affinity. Okay, you want to develop your own molecule with high affinity, then what you will do? Oh, hello? Hello?
Sir.
I'm audible?
Yes, sir. You are audible, sir.
So no, another approach is then comes that then comes in the picture your 3D pharmacophore modeling, or then comes your high throughput virtual screening. Okay, these approaches also you can use. Okay, high throughput virtual screening is very, very important if you don't have any lead in hand. If you don't have any lead in hand, then in that condition, what you have to do? Okay, you take the catalytic pocket. Okay, so there are two types of virtual screening. There are, I'll share it. 1 minute. There are two types of virtual screening.
Excuse me, sir.
Yeah.
Sir, have you shared the screen, sir?
Yeah.
But the screen is not visible, sir. Just now.
Yeah, I'll share it.
Okay, okay, sir.
By the time I'll share it.
Okay,
so
sir.
there are two types of. Okay. There are two types. Okay. We can say virtual screening approach. So one is, okay. One is, let me just open it file. I have to delete the old. I'll upload the new one. This these are the practical aspect that you're going to use. It's not very hi-fi thing. If you have software, you can use it. Okay, you can use it. Yeah, I'll share it now. Okay, it came. Yeah. Now, virtual screening, it may be, first I'll come to the structure base. You have the protein. You know this is the protein. You can go for the virtual screening. You can use the virtual screening and then you can find out your lead molecule. So how to do this? Okay. So prepare the protein. Okay, correctly. Minimize the energy and select as a query and then select the ligands from the databases. Okay, from the databases. So if you select the databases, yeah, 1 minute. Is it visible? 1 minute. Yeah, this one, this file. Is it visible now? This table?
Yes, sir. Visible, sir.
Hello?
It's visible, sir.
Yeah. Now you can see. So what I have done, we did high throughput virtual screening. Okay, virtual screening, safe molecules. What is safe molecule? I'll come to afterward. Okay, we have selected you prepare the protein. Okay, prepare the protein, generate the catalytic pocket, and select the ligands or whatever the database compounds are there. You can develop your own library also. Suppose you are interested in the anti-cancer activity. So all what are the databases are there for anti-cancer compound, you can select and you can make your own library. Okay, you can make your own library. Or even the free libraries are also there, paid libraries are also there. So you can use those library, you can go for the screening. Okay, when you go for screening, it will give you the output. It will give you the output. Okay, these are the compounds having affinity for this protein. Having this will take time. It is the time consuming. It is the time consuming. Okay, so it will take time, but it will give you the result. It will give you the result. Okay, but here, so you can see these are the, now you can see this is CPA. This is the name, chemical abstract name. Okay, all the compound library, whatever is there. Okay, it is stored by this name. And the chemical compound where it is available, that library also source it will give. Now you can see here, this is the time tick. Okay, these are the, this is Acinex. Okay, time tick, life chemicals, enamine, Maybridge, spec. Okay, ChemD, key organics. These are the different databases of these compounds which you can use, for your high throughput virtual screening. When you are doing for the virtual screening, then how you are going to control it? Suppose it is giving 2000 compounds. All 2000 compounds may not be active. Okay, may not be active. Okay, and then how to select from those active compound from those. So this is your another thing. Well, how to control it. Now here I'm showing this. You can see here. Yeah. Now when you're doing it, okay, when you're doing it, then you have to look into the glide score, any score, docking score, whatever is there. Based on the docking score, you can arrange it. You can arrange it. Okay, based on the docking score. Now suppose this is the 8. Okay, this is the compound having showing a score -8 kilocal per mole. Okay, then you can say here, suppose it is your, I selected here, okay, few compound, but it may result in 2000 compound. It may result in 2000 compound. So how to monitor it? When you're going for the virtual screening, then another protocol, what you have to follow here, okay, you give written compound only safe compound. What is the safety then parameter? It should follow the Lipinski rule of 5. Okay, it should follow the Lipinski rule of 5. Okay, then you can see these are the rule it should follow. Okay, it should not be toxic. HERG, okay, rule of 5, rule of 3. These things you can use and you can filter the compound. You can filter the compound. So rule of 5, rule of 3, you can apply and you can filter the compound. It will give you the safe compound. Safe compound. And the thing, now you have to admit property, you can calculate. Admit properties means it is there on the website. It is there on the free is there. Free trial. Swiss admit is there. You can use the Swiss admit, load all your compound and calculate the admit property. Calculate the admit property. And now after doing this admit property, calculating it, then you have to see what are your pharmacokinetic profile. It should be favorable. If it is not favorable, then why necessary? Why to synthesize those compounds and waste time? Okay. So before going for the synthesis, okay, before going to synthesis or any work, you have to look into that. Okay, these compounds should be safe. Now I'm looking, these compounds are not, you can use CSRB also. Okay, there also you'll get the same result. Okay, the CNS activity, okay, it is not having CNS activity. Molecular weight is low. Okay, Lipinski rule, it is following Lipinski rule of 5. Okay, then, okay, you can say hydrogen bond donor, hydrogen bond acceptor. Okay, HERG channel. HERG channel, it is very, very important. It should not go and bind with the potassium channel of the cardiac muscle. Okay, if it is binding with the cardiac muscle, then definitely it is going to produce cardiotoxicity. Then you have to omit that. You have to omit that one. So if you go for these calculations, HERG channel, there is no activity, very good. Okay, permeability, okay, there's cacao permeability, it should be high. Okay, then only active transport will be there. Okay, active transport will be there. Then you have BB, so it is very low, so it will not cross the blood-brain barrier. Okay, and then it should follow the oral absorption. Then you have to look into the oral absorption. Okay, what is the percent oral absorption? Percent oral absorption should be high. Okay, now you can see here absorption is high. Okay, 186 and 87. So what are the compound you are getting from the high throughput virtual screening that you have to rigorously analyze by using this ATME calculation. So there are 2-3 softwares are there free. You can use and you can omit those compounds which are toxic, Okay, that toxicophore, sometimes it will come in the molecule. Those all compounds you can remove and then take only the safe compound. So switch ATME you can use one software. There are so many software. So the rule of 5, you can see there should not be any violation. There should not be any violation of the rule of 5. And the stringent rule is Jorgensen rule of 3. It is very stringent rule. There is no violation. There is no violation. That means compound is good. Compound is good, and there is no toxicity. Okay. But another thing very important here, you have to see how many metabolites will be there. Okay. So metabolites should not be more. If metabolites are more for that compound, whatever your compounds you are selecting, then definitely it is going to produce. Okay. See the number of metabolites. You can see here, this compound is showing 6 metabolites. There is a concern. Okay. So there's concern. Now here, I will see this is 5, 3, 4, 4. Okay. This is 4, 2, 1, 3. So number of metabolites should be less. Okay. Then the compound will be safe. We don't know what metabolites is there because at this level you are not going to study those metabolites, whether those metabolites are toxic or not. So better to avoid. Okay, number of metabolites, if it is more, then avoid those compounds. So 3 things you have to keep it in mind. Or 4 things. First is it should not have CNS activity. It should not have any HERG channel blockage. Okay, it should not produce more metabolites. Okay, oral absorption should be high. Okay, and metabolism also you have to look. What way it will undergo metabolism. Okay, so these are all things you have to keep it in mind. And if this is fine, then you can go ahead with the compound. Now, suppose you got compound. Suppose this compound. Okay, this compound you got. Now it is available in the ICDEX. No need to synthesize. You can purchase it and see the activity. Okay, see the activity. This is the best way. Okay, so just see the activity. If it is showing promising result, then a structural modification you can do. Then a structural modification you can do it. Now let me see this compound. Yeah, this is the compound. You can see. Is it visible?
Yes, sir. Yes, sir.
This screen is visible?
Yes, sir.
Yeah. So you can see this is the structure. So what are the possibility of structural modification? Okay. What structural modification? Then again you import this.
Sir, I think you have zoomed in. The structure was not visible, sir. Only that toolbar.
I'll share it again.
Yes, sir. Yes, sir. Thank you, sir. Now it is visible, sir.
Is visible?
Yes, yeah. Sir. Now it is visible.
Now you can see this structure. Okay, this is the structure. HTVS. You can buy this compound. You can test the activity. If it is showing promising result, go ahead. Okay, you can then you have to look into how it is binding because you have done the docking. Again, the same way. Okay, you generate the catalytic pocket residues. Okay, binding residues there. Okay, shape of the pocket. shape of the pocket and analyze the pocket, and then you can optimize this molecule within the catalytic pocket. Okay, within the catalytic pocket, you can improve the binding affinity. You can improve the binding affinity. This is very good approach. Okay, you can save time saving. Suppose okay, time saving and money saving. Okay, suppose if you don't test it and then abruptly you synthesize some other molecule, and okay, and those molecules, okay, if not showing activity, then the waste of time. So it is always better to test the molecule and go ahead with the design part. Go ahead with the design part. This is the approach most of the researchers are using. Okay, and it will give you good publication also. It will give you the good publication also. On what basis you have selected? I have selected this heat. Okay, I optimized this heat. Okay, I've tested this heat. This is the IC50 value. Okay, and this is the, suppose it is antibacterial. You can MIC value. If it is some animal model is strict, that value you can put. Okay, this will validate your work. Okay, this will validate your work, and then you can go ahead. Then most of the PhD work, okay, should be based on this. Based on this is the way. This is the way. Okay, you can. This is the one of the approach. This is the one of the approach, and very good approach, because you can save time, money, energy, and with less time you can come out with the lead molecule. Now here lead molecule you got. So this is the compound. Suppose it is showing good activity. That means unless toxicity is there, whatever the ADMET property profile is there, it is good. Okay, pharmacokinetic profile is good. That means it is a good candidate, lead molecule. Then how to optimize it? Then optimization you have to use different tools. So one I told you, catalytic pocket. You can analyze this molecule within the catalytic pocket. You can modify this molecule by different substitution, and then some of the group may not be necessary. Okay, not necessary. Smaller the molecule, better. Okay, smaller the molecule, better. So what are the extra things are there? You can remove. You can improve the binding affinity. You can improve the binding affinity. But whatever the molecules you are designing, okay, after designing, okay, docking and all, again you have to go for the ADMET calculation. See the toxicity. Okay, whether any toxicity is there, ADMET profile is correct. If ADMET profile is correct, there is no toxicity associated with those molecules, then very fine. You can go ahead. You can go ahead with the synthesis. Okay, you can go ahead with the synthesis, and your research will become very, very strong. It will become very, very strong. Okay, so this is one approach. Now suppose, okay, this is one approach. Second approach is, this is the structure-based approach. What I told you, this is the structure-based approach. Another approach is there. In the literature, for any target, inhibitors are reported. Inhibitors are reported. So suppose any literature, okay, take any literature, suppose 60 to 70 minimum, okay, inhibitors are there with IC50 values, or okay, IC50 values, or okay, whatever the biological activity is there, okay, you take that one, use those activities and structures, okay, to generate the pharmacophore model, 3D pharmacophore model. If you generate the 3D pharmacophore model, it will give you the shape of the, it will give you the shape of the molecule. This is the shape of the molecule. This is required for the activity. Okay, so this is another approach. The 3D pharmacophore model. When you are developing 3D pharmacophore model, one thing you have to keep it in mind. Analyze the structures. Whatever the structures you have, okay, selected from the literature. Sometimes literature, they will not reveal the chiral centers. Okay, simply they will put the biological activity. Okay, there is a chiral center in the molecule, but okay, they will not reveal that one, what type of enantiomer it is. Okay, whether the levo or dextro, whatever is there, they will not mention. They don't mention. Leave those molecules. Don't select those molecules for development of the 3D pharmacophore model. This is one precaution you have to take. Okay, and when you are selecting these inhibitors, okay, you look into that diverse. Okay, there should be a range of activity. Some of the compounds should be highly active. Okay, and there's some of the compound should be low active. If the range is not there, then also you are not going to get good pharmacophore model. So to develop the pharmacophore model, there is a technique you have to look into. So first point, remove all those compounds having chiral centers if that activity is not reported. Okay. Second thing, you check log 4 difference should be there in the activity. Activity you convert into PIC 50 value. Okay. And then see, okay, the difference should be 4, minimum 4. Okay. Then it will give you the good model. It will give you the good model. Then you divide the molecules into active and inactive. Okay, how do you divide? There's a how do you divide? There are so many techniques are there. Scrambling. Okay, why scrambling? Why scrambling is the best method? You can use the Y scrambling if the number of molecule is more. Okay, if the number of molecule is more, you can use the Y scrambling method, and then you can divide the whole set of the data into active and inactive. Okay, active. Based on the active compound, you generate the model. You generate the model. Then you have generated the model. So how to validate it? You have to validate also that model. Okay, okay. You have to validate the model. So when you are validating the model, you take some predict the activity. First thing, whatever the inhibitors you have selected, again you predict the activity of selected inhibitor. Okay, whatever inhibitor you have taken. Okay, you just predict the activity, see what difference you are getting. Predicted activity and actual activity. There should not be more difference. There should not be more difference. Predicted activity. This is your internal validation. This is your internal test set, whatever is there done. Okay, test set, whatever that's what test set is taken. Training set for development of the model, test set for the your validation of the model. So test set you just use and then validate the model. Okay, and see it is showing the same activity, or there should not be more difference. Well, okay, deviation should not be more. If deviation is not more, fine, your model is good. Second thing, we have to take some external molecules, external set of molecules. Okay, having different diverse set of molecules with the same activity, and for them also you test the predicted activity. So it should show okay correlation. It should show correlation between the predicted and your experimental value. So if the correlation is 0.9, then very, very good. Your model is very good. Okay. So this is the external validation. External validation. These are two techniques generally used. Okay. Most of the literature you can find they are using it. Internal, external validation. Third technique is the enrichment study. So what is the enrichment study? They are decoys molecule. Okay, decoys molecule means those molecule doesn't have any activity, no activity at all, no biological activity. So your active molecules, what are the active molecules you have taken? Okay, those active molecules you mix with that decoys molecule. Okay, so it is ratio is like that. Your 20% and 75%. Okay, you can take 25, 75. Okay, 25 your compound, 75. Okay, or even you can increase that decoy also. Okay, so suppose I'm having 10 potent inhibitor, so I'll go for the 200 decoys. We mix, and after mixing, you generate a data set, and that data set again you screen. Okay, screen. So your model should be able to find all the active molecules and not the decoy molecule, not the decoyed molecule. Then also you can say, yeah, it is fine. Yeah, my model, whatever I've generated, it is good. Okay. So these are the 3 steps, minimum 3 steps. If you follow, then you will get the good. Use this model. Okay. Use this model. So there are 2 ways. Okay. Pharmacophore based screening, and then is the shape based screening. Shape based screening. Anything you can do. Okay, shape-based screening is very fine. So if you put this as a query, whatever the model you have developed, if you put as a query and go for the virtual screening from the data set, from the library data set, so what will happen? It will return the compound having the same shape, having the same shape. Now the molecule will be different. Okay, and those different molecules again. Some of the molecules may show very high activity. Okay, then you have to validate with the docking and then binding free energy. Okay, binding free energy, you can validate those compounds. Okay, this is showing high binding free energy. Okay, negative values is very high. Okay, docking score is very high. Okay, so this then those compounds also you can select as a lead. You can select as a lead, and the structure will be different. The structure will be different. Those molecules also you can okay optimize in the catalytic pocket. You can improve the activity. You can improve the activity. So this is another approach. This is another approach. So shape-based screening, you can go ahead. Pharmacophore-based screening, you can go ahead and you can find out the new molecules from the data set. You can optimize those molecules in the catalytic pocket, and then you can binding free energy, docking studies, all you can perform, and then you can come out with a good molecule. Those molecules you can optimize in the catalytic pocket, and you can design new molecule. You can design new molecule. So this is another approach. This is another approach. So this is very promising. Promising means a result it will give, and it is very successful. It's very successful sometimes. Okay, if your model is good, it will model is good. If your validation is good, definitely it will return very good compounds. Very good compounds. You can save time. There are targets where inhibitors are not known. Okay, so in those cases, structure base is the best. Structure base is best. And suppose more inhibitors are known. Okay, then this ligand based approach you can do. Okay, pharmacophore modeling. Okay, you can do. Okay, shape based screening you can do, and you can find out the potent inhibitors. Potent inhibitors. This is a very good approach. This is very good. Either way you can use. Either way you can use. Now, when you are going for the another thing I'll tell you. When you go for the virtual screening, it may result in 2000 to 2005 compound, 2500 compound. How you are going to handle those 2500 compound? For that reason, there is another approach. I'll tell you here. I'll share another one. So here, one by one, I'll show you this. Then you have to go for there's a method called clustering. There's a method called clustering. You can go for the clustering. Okay. The clustering is nothing but the group of compounds. Suppose 2000 compound is there. I'll share it. 1 minute. So 1 minute. Just 1 minute. Hold on. I'm sharing it. Yeah. Yeah. Can you see this one? Hello?
Sir.
Is it visible?
Yes, sir.
Yeah. This is the clustering. Clustering the molecule. 2500 molecule. You cluster into, suppose 10 sets or 15 sets. You have to use different protocols, and you have to come to position how many clusters are important. Required here. Suppose 15 clusters are not required, 10 clusters are required, 20 clusters are not required. 15 is required. So this will come after trial. 2-3 trial, you will come to know. So that you divide these all molecules, 2500 molecules, into suppose 15 clusters. Now you can see these are the clusters. So this group you can see here. Can you see? Okay. So this is the one group, second group, third group, fourth group, fifth group. So how this compound is related, it is showing by this line. This compound, there's so many compounds here. This compound, this is linked with this compound. This is linked with this compound. Now what happened? Okay, it will cluster the compound based on the structure, diverse structure. So each group is having diverse structure, and that is very, very important. Okay, it is not like that you whatever virtual screening you have done, and all compound whatever you are taking having the same scaffold, having the same scaffold. There is no meaning. Okay, the scaffold should be different. Scaffold should be different. So scaffold is different, then it will give you good result. Now you can see here the different clusters are there. Okay, and in these all clusters, we have different scaffolds. Different scaffolds. Now, these clusters, okay, already you have the score. So from each cluster, you can select five compound from each cluster. So 15 cluster is there. So total it will come 75 compound. Okay, the 75 compound you select, then what will happen? You have the diverse structure. Your representation is with diverse structure. Now, if diverse structure is there, that is always good. That is always good. Those 75 compound, you can go for the docking, you can go for the binding free energy calculation, molecular dynamic calculation, simulation studies. This all you can do it. So this clustering is also very, very important. Here the photograph I show you here. 1 minute. So this clustering is also important. Okay. Now, yeah. 1 minute. Is it visible?
Yes, sir.
Now new. New share. I am doing this one. Is it visible now? Kelly's penalty. Is it visible?
Kelly penalty is visible, sir, now.
Yeah. Now you can see here.
Yes, sir. Yes, sir.
So based on this Kelly penalty, there are two ways of doing this clustering. Okay. So there are two ways you can so you can do it by any way. Okay. So the number compound is more. Okay. Then you can. So that's this. There's so two ways are there that you can do. Okay. K means and your another other method also there. So here you can see here this is the minimum penalty is Kelly penalty. It is a minimum penalty. Penalty means minimum at what position? So here you can see, okay, that it is showing around six cluster or 7 cluster. So you cannot go beyond the 6 cluster. If you're going beyond 6 cluster, then what will happen? You will not get the good clustering. You are not getting a good clustering. So this is also important. You can judge, okay, your clustering method, whether it is good or not. Okay, it is good or not. This is one of the things. Okay, Kelly's planting. You can judge your thing. Okay. Now another thing here. One more thing I'll tell you. Yeah, distance matrix shown dendrogram. Yeah. 1 minute. One more thing here. There's so many. Okay, there's so many things are there. So you have to validate that. Anyway, whatever the clustering you have done, you have to validate that. Then only you can say, yeah, it is good. It is good. Now you can see, okay, on the scale, cluster order. So this will also tell you, so which is good. Hello? Yeah. Yeah, here you can see, this is the red region, this is the yellow region, this is the blue region. So this all it's falling on the line. So hello. So now here, if you look into these points, you will come to know what are the compounds falling on this one. Okay, this compounds very nicely you can select. Okay, the clustering method is also very, very important. Very important. So you understood this one? So this is the virtual screening approach you can apply. Then lastly comes, okay, binding free energy calculation. And then lastly, you have to do that. You have to validate the docking, okay, by molecular dynamic simulation. Molecular dynamic simulation. So because time is not there that much, I cannot go in more detail. So in short, I'll tell you how to go ahead with that. Okay. So I'll share another presentation. How when you're doing the molecular dynamic simulation, so what are the things you are going to, okay, you are going to follow. One minute, let me share it. It will be useful for you, okay, when you use software, any software, free software, So it will be always useful for you. Is that visible?
Yes,
Hello?
sir. Yes, sir.
Yeah.
Yes, sir. It's visible, sir.
In the molecular dynamics, okay, in the molecular dynamics, the molecule is described as a series of charge point atoms linked by a spring. Linked by a spring. So there is always, I've shown you the photograph earlier. So it is in constant movement. Bonds are stretching, bending, okay, stretching, seizing, all this is happening, okay, during your binding process. So in the molecular dynamics, you will describe the time evolution, okay, bond length, bond angle, torsion, non-bonding, van der Waals, and electrostatic interaction. Okay, these are very important. These are very important. All the force field, anything, anyway, you are going to use the force field, all the force field may not calculate, okay, non-bonding van der Waals interaction. Okay, some of the, then you have to look into what force field you are using. You need this one. This is the dipole-dipole interaction. This is the dipole-dipole interaction. So this dipole, because protein is a very big molecule, so there's always, you have dipole-dipole interaction there. So if you have dipole-dipole interaction, so you need to have van der Waals interaction. You need to know how much it is contributing. Okay, when you go for this, now you can see here, this is the function of time. Okay, analytical solution of equation of motion to give time and velocity is impossible. So we use this one, okay, trajectory. We generate trajectory of all particles in the system as a function of atom is moving in the system. For this, what we do? Motion the system through space is governed by Hamiltonian equation of motion. So we apply here Hamiltonian equation of motion. Okay. Now, yeah, when you go for the solvation, anyway, when you're going for the molecular dynamic simulation, you are solvating. You have to be careful. Okay. When solvating, it may be either implicit, it may be explicit. It may be either implicit or explicit. Implicit means, now, explicit, each solvent molecule is considered. Here, this is applied in case of liquids and solids. Okay. Liquids and solid, explicit system. Implicit system, solvent is treated as a continuum description. This is, you can see this diagram, continuum description. Okay. So need to define a cavity that contains the solute. Here, this is the cavity. Okay. That contains the solute. Suppose one solute molecule is going on, another will enter. Another is, so there is a continuous movement. Okay. The number of, but the number will be same here. Number will be same here. Generally, this implicit system is used. Generally, implicit system is used. And when you are applying, okay, when you are applying this, then another thing you have to keep it in mind, okay, cutoff methods. What are the different cutoff methods you have to apply? Short range coupling will be there, long range coupling will be there. Short range interactions, okay, short range interaction, okay, cutoff radius, okay, for pairwise interaction, calculate the potential use of for those within the cutoff space, okay, within the cutoff space, okay. So this you have to define cutoff space for interatomic distance, okay. Then electrostatic interaction is a very strong and falls very slowly, okay. So these are the long range interactions, long range interaction. So generally, to treat this your electrostatic interaction, we use, okay, almost all simulation, whatever is there, software you use, evolved simulation, particle-based evolved, okay, they use it, okay. So this cutoff also is treated there, okay, electrostatic, okay, interaction, for van der Waals interaction. Okay, these are treated here. Now, what are the model? There are different type of model for the dynamic simulation. So, which model you are going to use? Nowadays, newer one is the TIP4P is always better. TIP4P is always better. Earlier it was TIP3P. So here TIP3P you can see, okay, three sides are Q1, Q2, Q1, Q1, Q2. Okay. Yeah, 4P you have, okay, Q1, Q1, Q2, okay, and plus this. Okay. So there are four sides are here. Here it is the five side. Here it is the five side. Some of the companies for the metallurgical study they use TIP5P. Okay. So water model with 3 to 5 interaction site. For our work, TIP4P is always better. Now you can see why it is better. You can say, no, water, okay, in solid phase. Water exists. Okay. Okay. Most complex phase diagram. It's showing most complex phase diagram. Having 13 different solid structure. Okay. Now you can see here. So this is TIP4 model. Okay. This is the diagram. This is the diagram. Okay. Temperature versus pressure. Okay. Now here you can see this is the experimentally determined. Now you can see this is very near to each other. It's very similar. Very similar. But when you take the older model like SPC or TIP3P, it is deviation is more. Deviation is more. So that means okay, your TIP4P model is better compared to the SPC or TIP3P model. Okay. So water model, you can select the TIP4P. Then when you are going for the selecting that, there are so many okay variables are there. Variables are regulated concept. Okay. So selection of ensemble. You are running the dynamic simulation in ensemble. It may be constant energy, constant value, one constant temperature, constant volume. Okay. It may be constant temperature, constant pressure. Okay. So this is the best one. So constant temperature, constant pressure, if you maintain, this is always. Okay. So NVT generally, sometimes they apply for equilibration. They use this one NVT, then go for the NVT. Then go for the, suppose you are running 100 nanosecond dynamic simulation, go for 5 seconds, 10 nanosecond for the NVT, then go for the NVT. It will give you a very good result. Okay. In this dynamic simulation, your integrators are there, so that pressure, temperature, everything is controlled. Okay. Everything is controlled. So when you are running the dynamic simulation, okay, so temperature, pressure should not change. Okay, it should not change. And then when you generate the data, then it will give you the accurate figure. Now I'll show you one result here, dynamic simulation. Okay, so here 1 minute. So it is very good. Here, one result I'll show you how important it is. Okay, it gives accurately what are the different interactions. Okay, what are the different interactions? Now, is it visible?
Yes, sir. Yes, sir.
Now you can see this is the inhibitor. Okay, inhibitor. After dynamic simulation with the protein, it is showing 14 RMSD. Now during dynamic simulation, you have to look into C alpha should not change. I told you earlier. Okay, C alpha carbon. Now what I'm looking here, the C alpha position, this is the blue color. So it is C, it is coming, but it is stable after. You can see it is stable here. So this validates your dynamics. C alpha is not changing that much. So protein flexibility is less. One thing. Second thing, okay, RMSF. Okay, root mean square fluctuation. So here, root mean square fluctuation, only see maximum it is low. Maximum it is low. Okay, so that is also one of the things. Third thing, you can see here, important thing, how it is binding with the inhibitor. Now you can see, so what are the different interactions are it is showing? It is showing the hydrophobic interaction. It is showing the hydrophobic interaction. You can see this is hydrophobic interaction. These are the hydrogen bonding. So most of the dynamics simultaneous every time it is showing, okay, hydrophobic interaction. Okay, and this is your water-based interaction. Hydrogen bonding, water-based. Okay, hydrogen bond is very less important. Less important. So this information you're getting, and then plus, okay, another thing you see here. Now, another thing. You can see this inhibitor, it is showing the rotation. Single bond rotation. There are so many single bond rotation here, isn't it? You can see rotation here. So this is the all the rotation due to rotation. Okay, what will happen? You can see this is the total rotations are there. Okay, 360 degree, 182, minus 180. Okay, there, this is rotating. These are bonds are rotating. The blue color, green color, you can see it is rotating. Green, it is rotating. How to avoid this? If you want to improve the activity, then you have to prevent the rotation. What will be the solution here to improve the activity? No, very simple thing. Just connect these two carbon atoms. If you connect these two carbon atoms, all the rotation will be prevented, isn't it? Because cyclic structure, okay, rigidified structure. If you rigidify the molecule, definitely rotation will be less. It will be more stable and it will show more activity. Okay, so this much information it is giving dynamic simulation. You can design the inhibitor based on this result also. Okay. Okay. Now this time is getting over. So any question? So hello, my time is getting over.
Hello, sir.
Okay.
Yes, sir. That was a wonderful session, sir. We have some question in a chat box, sir. Can you access or let me read out, sir?
No, this master software I'm using, this is not the licensed version. I have downloaded from the site, okay? Academic version. Okay? This work I have done some other places. Okay? Okay? So this is one.
One more question was there, sir. Can molecular dynamic simulation can be done in laptops?
No, you cannot do in the laptop. Never try. Okay? Never try. It should be on the desktop and then to the Linux system. Linux. Okay?
Okay, sir.
In your
with Windows we cannot.
Okay?
Okay.
Never try to run the molecular dynamics in word processor. Okay? Okay? Okay, Windows you don't try. Windows you don't try. Okay?
Okay, sir.
Only
With
the
Linux only we can.
Okay?
Okay, sir.
A Linux system is always better.
Okay, sir. Okay, sir.
Okay? Okay?
Okay, sir. Sir, actually, Niranjan Babu sir was in inspection, sir. One inspection is going on in college, so he couldn't able to join with us, sir. Sorry for that. Sir will talk you with later, sir. By evening, like sir will call you, sir. And I'm taking this opportunity to thank you, sir, within a token of gratitude. Can I stop your screen share, sir?
Yeah. I just stop sharing the thing and then I log off. Okay?
Sir, 1 second, sir.
Okay.
Just accept our token of gratitude, sir, with a memento and one certificate.
Okay.
And the same will be posted you, sir, with the courier.
Okay. Thank you.
This is the certificate, sir. Thank you so much, sir, for joining with us.
Okay.
We're expecting your
continuous
support to us.
Thank you all the delegates for patient hearing. Okay, patient hearing. Okay.
Thank you so much, sir.
Okay.
Yes, sir. Dear delegates, that's all the scientific sessions for day 3 of an AICTE sponsored ATAL-FDP on artificial intelligence in drug repurposing in polypharmacology. So tomorrow we'll meet at by 10 a.m. sharply for the day 4 sessions. I hope you will have the continuous scientific sessions tomorrow too. And the feedback link for the today's session will be shared in the WhatsApp group by evening, and kindly do it by today itself. And thank you, dear delegates, for the continuous support to us. Thank you so much. We can leave the meeting. Meet you tomorrow by sharply 10 a.m.